more attention to maintain the culture conditions. Now, there are some things like in vitro, in vivo, ex vivo, in silico. The four terms are very commonly used in the medical research. In vitro, just outside the body to carry the cell from the organism and put into the any petri dish or the medium, it will easily grow. Then there is a in vivo, that means you are injecting the drug into the animal, like animal models. Then ex vivo, if you are performing the dose response curve like DRC, you are getting the tissue from the animal, then apply outside in the assembly, maintain all the condition and do the experiment. And then fourth one is in silico, I will discuss here. Now, the one of the common example, which is a very old example, that was the hen's egg test was used. So here I will show you that this is a full thickness skin model. Now, this outer layer of the egg was used to apply the cream formulation, so that just we apply over our skin. So whether to check how much concentration of the drug goes through the pore of the membrane to the inner side or absorbed from the skin membrane. So this was the full thickness skin model. So there are some drawbacks of this model, because this is not exactly a same like our membrane, but because our membrane is having a specific pore size. If you see in the blood brain barrier, pore size is 4 angstrom. If you see the outer peripheral skin layer, which is formed of squamous epithelium, it is having a pore size around 30 to 40 angstrom. So that means little bit variations are there. Next are the in silico. This is completely a computer based. You have to work on the computer. No need of media, no need of cells, no need of animal. All the things you will avoid here. But yes, you will use a computer along with a particular software, in which firstly you will draw a structure of protein. I am giving an example here. If you want to see the effect of dopamine agonist over the dopamine receptor, so you should know that there you have to create a protein which is having a structure of dopaminergic receptor. Then you will bind your new ligand over there, and then there you will see the interaction, you will see the energy minimization gap, so that you will analyze from these things. Another are the cell lines. So I have discussed already that there are some primary culture. The If you take the cell from the organism, grow in the medium, again take the cell from the first petri dish, move into the second petri dish containing medium, that is the secondary culture, and again isolate the specific type of the cells, they are called as a cell lines. Now, cell lines, they are basically of two types: finite cell line and continuous cell line. So finite means cell line with a limited culture lifespan. If you have purchased the cell line, you have keep the cell line into your laboratory, and the cell line can be damaged after 3 days, 4 days, but not survive more than 15 days or 10 days like that. They are finite type. So that means shortly you have to use, and then you get your results. Then there are second type of the cell line, which are called as a continuous cell line. So that can survive for a long time. It can be for 20 days, it can be for 30 days. Again, question is here that how the finite are surviving only for 1 days, 2 days, continues for month or a 20 days. Now this depends upon the environment of your lab also. That means if you have not completely fumigated your lab, अगर वहां पे कंटेमिनेशन के चांसेस बहुत हैं, तो ये चांस रहते हैं कि आपकी सेल लाइन में भी डिग्रेडेड, द सेल लाइन में भी डैमेज. ये डिपेंड करता है आप पे कि you are using a gloves, you are using a proper mask, you are using a head cap. That means you are maintaining the sterilized area there. So that are the things to be maintained the cell line for the long time. Now, these are the common property of the cell line. What is the difference? That means growth, finite, slow, continuous cell line are fast. Then mode of growth, it could be monolayer suspension. Now, if I discuss here that several students have joined this presentation, that the culture as well as the cell line could will not be a solid medium. It's not a solid medium. It is either a semi-solid and it is a liquid medium. So it will either a suspension form, it will either be in the emulsion form, or it could be in the liquid form itself. Now, what is the yield? Finite low and continuous they are high, because continuous are more specific. वो एक सिंगल सेल की होती हैं. अगर आपको स्ट्राइएटल D1 डोपामिनर्जिक न्यूरॉन का सेल लाइन चाहिए, तो दे आर स्पेसिफिक टाइप. So that means there will be not presence of any other type of cells in that. So then is the transformation, cloning, serum requirement, markers. So these are the difference. So what are the applications of the cell lines? Now, the major applications of the cell lines include screening of anti-cancer drugs. I have told already that no protocol for the cancer study is being approved by the Institutional Animal Ethical Committee. The condition, you cannot do the cancer in the normal human being. You can do the cancer in the animal also. So that's why for the anti-cancer drug testing, the phase 1 clinical trial is not there. You cannot use the healthy volunteers for doing the cancer studies. Second is the cell-based bioassays. Next, to determine the cytotoxicity, this is a very simple thing. Just you take a concentration of the culture into the petri dish, 3, 4, 5 petri dish, put the different concentration of your drug, 10 microgram, 20, 40 in the sequence. Then you do whether the cells are surviving or not. This is a simple method. In vitro screening of several drugs, production of antiviral vaccines, cancer studies, cell fusion techniques, genetic manipulations, gene therapy, rDNA technology, biotechnology, and molecular biology. So next important technique is a patch clamp technique. So I think so. Maybe many of the people researcher are aware about this, and maybe many not, because this is the patch clamp method. And just I will give here the example of HERG analysis. That means human ether go gene, the gene which is responsible for the conduction of potassium ion into the cells of the heart. That means the cardiac cells. That whether it is required, why the pinch clamp method is used. Just you can see the movement of the ion across any cell. The cell could be neurons, cell could be a beta cells of the pancreas, cell could be any muscle fiber. So how the amount of the sodium, potassium, or the calcium, these ions, they are moving through the channel across the cell membrane. So this can be tested by the patch clamp. I will show you in figure. So this is the figure of a patch clamp. On the lower side, you will see the cell is there. There is outer membrane. On that membrane, the ion channels are present. And after then, from the upper side, the electrode is also supplied along with the connected with the cells, and the electrode which is present inside the pipettes. So that means the pipette in which the electrode is present, this is not the full diagram. This electrode which is showing on the upper side, this is further connected to the amplifier resistance and then to the receptor side. Now what will happen? This pipette contain a solution mean if these green channel, I am giving an example here, if these channels are potassium channels, so that means you will connect a channel by the pipette which is present, which contain a electrode. Now this pipette contain a solution of maybe potassium, sodium, calcium, or electrolyte solution is present in the pipette. Now what will happen? The ions always move depending upon the pressure across the cell membrane by the method of diffusion. Now whenever the outer side potassium is more, it will start to move on the inner side. That you know that on the heart there is a presence of sodium potassium ATPase channel. That means the movement of the ion across sodium inner potassium out, and likely it can be transferred on the heart membrane with the potassium with the calcium also. Now what happen here, you know what is the concentration of the solution in the pipette. ये आपको पता है कि पिपेट में कितनी कंसंट्रेशन है पोटाशियम की सोडियम की, बिकॉज वो सोल्यूशन आप लोगों ने खुद बना रखा है. So you know the concentration. Now whenever the potassium ion through the potassium this green channel start to move toward the inner side of the cell, the voltage will be built up, the electric potential will be built up on that side, and that electric potential will be analyzed by the help of amplifier and resistance. That means, so how many potassium, what is the current of the potassium ion, what is the charge, what is the electrical voltage created by the potassium ion over the cell membrane, you can analyze. Similarly you can analyze for the sodium, for the calcium, or likely that. So this is a very important thing, because whenever we go for the toxicological testing, not a new drug tested only for the a single compartment model, it can be tested for the not for the single organ system, it can be tested for the multi organ, it can be tested for the genotoxicity, reproductive toxicity, CNS toxicity, or cardiovascular toxicity also. Now see, there are so many drugs. Similarly, it was the derivative of the cinchona, that means quinine and quinidine. So that is the difference between the stereo and dextroisomer. So one was responsible to cause arrhythmia. Now why the arrhythmia was there? That was interfering with the channels which are present on the cell membrane. Now see, if there is any newly invented drug that interfere with any of the channel on the smooth muscles um, on the any of the cells of the body, that means it can alter the physiology of the cells. So for this the patch clamp method was developed to analyze the electrical voltage created by the particular ions. So type of patch clamp, it depends that where you are having a connection with the clamp on the cell, inside the cell. It could be whole cell if the cell membrane is open, along with it could be outside but connected with the cell membrane. So now this is the principle. Similarly, I have told you that in the kidney help to transport a number of ions and help to reabsorb the ions or the molecules which are required to the body. So that's why ionic composition is not stable in the cell at all the intervals. It changes always, because whenever you take a food, whenever you drink a lot of water, the ionic composition will be different. But if you are having a lot of thirst, the ionic composition could be varies in the body. Now this is the thing. So this is the procedure I have discussed with you. They have used the segment of the late superficial proximal tube of the rabbit kidney. Now they analyzing the movement of ion across the proximal tubule. They have taken a part of the tubule actually, and the tubule part from the one side is connected with the system, and the from the another with the perfusion system. That means the solution move from your pipette side and go toward the perfusion side. So they again analyze the movement of the ion through the membrane, through the cells. So this is the evaluation. So what are the applications of the patch clamp technique for the evaluation of antiarrhythmic drug? I have already discussed with you that there are chances that many drug can cause arrhythmia. So it is useful for the antiarrhythmic in the kidney cells to analyze the composition, ionic composition in the ventricular myocytes. You can take from the animal to identify the multiple types of the calcium channels, as well as potassium channels, as well as voltage gated sodium channels, as well as to investigate the electrophysiological properties across the cell membrane. So this was about the ethics. This was about the uh, IAEC. This was about the alternatives. Now I am going about the animal house, which is present in the ISF College of Pharmacy, Moga. So this is the gallon animal house that is approved by the CPCSEA for the breeding as well as trading of the animals. So on the lower side, I have mentioned here the registration number of our animal house. So this is the entry door of the animal house. And then there is a dirty corridor. I have written on the lower side of the slide that the dirty corridor is there, that whenever a person enters should wear mask, should wear surgical cap, or what are the requirement if you want to enter. Then there is a you are looking on a switch are present there. That means there is a regulation of 12-hour light and 12-hour dark cycle in the animal house, because this is the condition of the CPCSEA, that you should be having a 12 hours light and dark cycles in the animal house. Because if you will completely switch on the light for the 24 hours, the animal can go under the stress. But if you will completely mean switch off the light, again there is a problem. But I will just tell you for the knowledge, the animals always breed more. Once you switch off the light, animals always take a more food whenever you switch off the light. This is our analysis that I we have predicted. Now this is the board where we have written that if anybody need the animal availables are Wister rat, we are having a Wister rat, albino mice, we are having a C57 bulb J mice, that is a genetic breed also. So these type of species are available. We also supply to so many university and institutes also. Now see what is the function of the dirty corridor. In the next slide, you will see there is again, this is a dirty corridor. Now in this dirty corridor, if anybody want to go inside the animal house, want to see the animal from the outside, he or she can move into the dirty corridor. And if you see the door of the animal house, the mirror is given on the in the door side. That means you can look into the room from the outside, but there is a no permission to go inside the room. So you should wear all the apron, surgical cap and everything, and you will, this is according to the CPCSEA, this is again according to the guidelines of the CPCSEA, that there is a dirty corridor on the outer side and there is a mirror in the door of the animals. Now, the animal rooms are again segregated depending upon the guidelines of the CPCSEA. So there will be a quarantine area, and then there will be a experimental area, then there will be a stock, then there will be a breeding area. So likely that whenever you will bring the animal from the lab side, when will you will purchase the animal from the other sources, so animals are always be quarantined. Similarly in the corona problem, you know that if you are having any chances, then you are quarantined by the government for the number of days. So animals are quarantined here. So accordingly we have given the shape to this animal house. So this internal is the clean corridor. Now outer was the dirty corridor as per CPCSEA norm. Now this internal is the clean corridor. In this clean corridor, only a person who is the scientist in charge, who is the in charge of the animal house, who is any nominee if coming for the inspection, any chairperson of the institute, along with maintaining all the conditions required to be entered in the animal house, are to be allowed to enter this in the clean area. So you can see the animals, you can judge what is the if there will be any kind of inspection or like that. So this is the internal clean corridor. On the both side of the clean corridor, that is closed by the door, but again you see all the doors contain a mirror. So that means this is very important part. No, this is the stock area where you see all the animal boxes. They are labeled with a particular tag, and in the tag we have written that how many animals are there in a single box, whether they are male or a female, because stock room always separate the male from the female. So this is the internal area. So next part, which is very important and going on in our institute from the last around 3 years, that is the ecotoxicological laboratory. That is the zebrafish strain Tenurario we are having. You can below see we have placed the fish into the drum, and that is the zebrafish. You know that many of the people they ask several times why it is called as a zebra, because zebra is a big animal, how you have placed here into the drum. But you see the stripes are present over the body of the zebra, the strips, white color, black color, the rows. Maybe if they any person who are here joined this, you see similarly if you see the outer skin of this zebra fish, these stripes are present which are having a black color, light white color. So that's why on the basis of that, the name of the Danio species is given as a zebrafish. Now this is a very small species. You can see having a total length of around 1.5 to 2 centimeter. Never we go beyond than that, with a body weight in only a mg, which is a which is around we can say 40 50 around. And then a small species which easily be retained in your laboratory. It is not difficult to breed, it is not difficult to maintain in the laboratory, because just they required a little amount of feed. So that means this particularly zebrafish is a very cheap research tool if you want to do the research on the zebrafish. Why? Because first thing, it is easy to maintain in your lab. It do not require a large area, and there are no specific CPCSEA guideline that you should be having a big house in which you can maintain the zebrafish. Only you see the tank which contain around 200 ml of water, 2000 ml of water, and in which you can place around 2 to 300 of the zebrafish at a time. So little bit feed they required, and just to maintain them you put the water, you add a some amount of the sodium chloride, because these are the sea fishes. Actually they are that the strain which is breeded from the fishes, and that need the sodium to be there in the water, that is required for the breed also. So this zebrafish is a good tool for the drug testing, for the chemical, pesticide, nanopharmaceutical, as well as the nutraceutical testing. And now this zebrafish in our ISFCP we are using for the neurotoxicity testing also. So we are validating the model of Parkinson disease, and just a year back we have also validated a model of epilepsy by using the zebrafish, and there was a good response of epilepsy by the zebrafish as compared to rat and mice, that I have personally analyzed. So this is a good species if you want to validate the neurological models. Then you can do the pesticide toxicity, carcinogenicity, and currently some of our students they are doing the experiment of the zebrafish by using the acrylamide induced neurotoxicity, sleep deprivation. So again, the thing is that for which kind of the experiment, for which kind of the research you required a zebrafish, or you required a rat or a mice or a guinea pig. Now if you want to do the research related to the sleep deprivation, so these are the good models. So you can use these animals for the drug testing or for this type of the research. So just a quick recap on these all. Animals are necessary to be used for the safety, for the drug testing, considering the limit of animal utilizing for the, so either you should having a limit to reduce the number, or alternative should be followed. National international guidelines should be followed. Test drug only be used if you are having a specific model with the clinical perspectives. Now third, last one, teaching and knowledge. So you should know that you should be having you should teach to the young researcher about the ethics which are required for the handling of the animals. So next, put the kind back in the mankind. So this is the for the animals. So this is all about the presentations. So I welcome if any of the question is there, any discussion will be there. So thank you very much for watching this lecture.
Thank you very much, Dr. Shamsher Singh Bajwa ji.
Thank you.
We thank you for your actually words are limited because the presentation was a comprehensive one. You have covered each and everything related to the CPCSEA, and I must really congratulate you as well as
I have not discussed the CPCSEA. Just I have just showed you the outer framework of CPCSEA. That CPCSEA
no,
will be fully explained by the second speaker.
Definitely, sir. Actually, what I meant to say was that you have actually, I must congratulate ISF and you also because your animal house is such a beautiful facility, and I'm really glad that we are able to see it from far off. I'm speaking from Bhopal, and really we are very glad that such a fine animal house is being maintained by ISF Moga. And sir, because of the technical glitch, we could not present your brief introduction that I would like to present now. So I request you to kindly stop the screen sharing so that Dr. Nilesh Jain will share your certificate, and I'll read out a small brief introduction of yours. And definitely after that we'll take up a few questions because already Thakur Gurjeet Singh sir is already online and he's waiting for his lecture to start. But we still have 2 minutes to spare. So I request Dr. Nilesh Jain to please uh, share the certificate so that I could read out the citation of Dr. Shamsher Singh sir. So Shamsher sir, since we are not connected physically right now, so this is the only method by which we can present you the token of appreciation from the Sagar Institute of Research Technology and Science Pharmacy. And to my August gathering, let me give a brief introduction of Dr. Shamsher Singh sir. He is presently working as an associate professor and head at Department of Pharmacology, ISF College of Pharmacy, Moga, Punjab. He did his pharmaceutical education, M.Pharm and PhD from ISF College of Pharmacy, Moga, which is affiliated to the Inder Kumar Gujral Punjab Technical University, Jalandhar. And he is contributing dedicatedly towards pharmaceutical education and research from the last number of years. He is a prolific writer with more than 35 publications in various reputed peer-reviewed journals to his credit. Besides this, he is contributing to almost 15 international and national book chapters related to experimental animal modeling and role of neurotransmitters in the CNS in the reputed journals like Springer, Nature, and he has also contributed 2 books. He has guided more than 22 students in UG and 4 pharmacy doctoral, that is PharmD students are under his work right now. 17 M-Pharmacology students are working under him. And he's also guiding 6 PhD students right now in the area of neurological disorders like Parkinson's, Huntington, Alzheimer's, etc. He has also been instrumental in having 1 patent from the government of India. He has traveled widely and talked about on various stage and platforms across India, and he's also presented his research work in Malaysia, Vancouver, and Japan, and received more than 5 international grants from the Movement Disorder Society and other organizations. He's also presented his research works at national and international platforms and got more than 6 Best Poster Awards, Oral Awards, and other awards. And he has also got an award from Punjab University, Chandigarh, IBRO Symposium. And he got second Best Paper Award from School of Neurosciences, University Technology, MARA, Malaysia. And he has also been awarded with gold medal and first oral presentation award by University Center of Excellence in Research, UCER, Baba Farid University of Health Sciences, Faridkot, Punjab. Sir, SIRTS Pharmacy is honored and glad to have you as one of our revered speakers. Thank you very much for your presentation. Now, I would like to unmute a few students who raise their hands if they have any queries, and then we can proceed with our second session with Dr. Thakur Gurjeet Singh, sir. So I'm going to unmute now Nikhat Farhana Ansari. She has raised her hand, and Vishal Kumar. Please ask your question.
Hello, good morning, sir.
Good morning.
Hello, good morning, sir. Thanks, it's a nice presentation. Actually, I am applying for the grant. For that, I am having animal study. So what I want to replace that animal study with the in silico method. Sir, we have to purchase the software or any free software, sir, were available to do the
So,
experimental
so
develop?
firstly, the thing is that you are applying for the grant. That is the secondary thing. And if you are shifting the animal study for the alternative method, that is also your interest. But I will just tell you that if you want to do the computer related software, that you can purchase, that there are some software that are available. You can search on the website also, that you have to purchase, like the gold score is there, that will be purchased for the docking study, and that is, I think, costly also. That is having a cost for the 1 year is around 10 lakh. Another thing, if you want to do without purchasing any software, you can also do, because free of cost, that means for the free time, just for a 1 month or a 2 month, the free trials are available, so that you can register there and you can use that type of software just for a 1 or 2 month only.
Sir, name of any that kind of software?
That is the gold score.
Oh, okay, sir. Thank you, sir. Thank you. Thanks.
Thank you, Nikhat.
Thank
Now, Mr. Vishal Kumar, you can please be brief. Ask your question, Vishal Kumar, and Miss Monica.
Good morning, sir.
Good morning.
Sir, my question is that how can we select anesthesia for the dissection? Like if we want to do stereotactic surgery, and particularly for a specific organ, how can we select?
Now, your question is very good. The question didn't depend that you want to use the stereotactic surgery. The question depends that which type of the research you want to go for. Now, I will give you example. Suppose if you want to do the research by using the calcium channel blocker. Suppose if you want to use a calcium channel blocker, so that means on the other side, if you are using an anesthetic, which is again a calcium channel blocker, so that will create a problem, that will not give you a good result. Okay.
Sir, one more question.
Okay.
Sir, if we replace animal study to in silico study, so there is any difference between results?
Yeah, there are some mean things in which you can change. You can go for the alternative method. Not at all. I have told already that if you want to see the physiological condition, अगर आपको ये लग रहा है कि मैं ड्रग दे रहा हूँ रैट को, और 10 रैट थे स्टडी में, या 6 थे, या कितने भी थे, दे आर गोइंग अंडर डिप्रेशन, तो सॉफ्टवेयर आपको ये नहीं बताएगा कि उसको डिप्रेशन कितना हो रहा है. सॉफ्टवेयर आपको ये नहीं बताएगा कि क्या उसको मसल ट्विचिंग हो रहा है या नहीं हो रहा है. ये नहीं बताएगा कि उसका टेम्परेचर बॉडी का बढ़ रहा है या कम हो रहा है. सो लाइकली दैट यू शुड नो दीज थिंग्स.
Thank you, sir. Now, since we are already running short of time, Miss
Okay.
Monica, please ask your last question. After that, we will hand over the stage to Dr. Thakur Gurjeet Singh.
Hello. Good morning, sir.
Good morning.
Sir, I'm asking about CPCA guidelines and protocols.
Okay.
What is the difference between CPCSEA guidelines and protocols? क्या differences होंगे सर इसमें?
They particularly, I think so, the topic will be more clear if you attend this second lecture, but I will give you some viewpoint about that. Now, if you want to initiate any kind of research, that means you are designing any kind of research, you should know that what are the area, what are the particularly the things in which you cannot go for. No, the first thing for the CPCSEA, if you want to sacrifice the animal just to get the organs, if you want to sacrifice the animal just to get the organs, to see the organ toxicity, then you go for the culture, you go for the cell culture. So no need to extra sacrificing the animal. And the second thing, if you want to analyze the effect of the drug, which is an antimicrobial drug, now why there is a need to cause the microbial infection to the normal animal? CPCSEA didn't allow. So there are so many things. I think so this will be more clear once you see the speech, listen the speech of Dr. Thakur.
Thank you, sir. Thank you very much for this. I have another question, but I'm sorry because since we have important time of Dr.
You are welcome. You are welcome.
Dr. Thakur sir is actually Thakur Gurjeet Singh sir is waiting for a long time now.
Welcome to Thakur sir.
Sir, I'll unmute you, and meanwhile I'll call upon my co-host Ms. Payal Saiju ma'am to please come up and present a small brief of Dr. Thakur Gurjeet Singh sir, so that we may plunge into the second session.
First, thank you so much, Sukhman sir. And first of all, I would like to thank Shamsher sir for spending time and giving us so much valuable information. And now we are entering into the second session of the day. And for this, we have with us Thakur Gurjeet Singh sir for this session. And I'm here to introduce to Gurjeet Singh sir to all our participants. Sir, on behalf of SGI family, my director, Dr. Jiten Banveer, and all the participants, I welcome you for this session. Sir has a vast experience of 13 years, and presently Sir is working as a head of Department of Pharmacology in Department of Pharmacology in Chitkara University, Punjab. He has a comprehensive knowledge of basic principle of clinical and preclinical development, drug development, derived from extensive graduate training in pharmacology, and he did his PhD in 2016 in Faculty of Pharmaceutical Science from Chitkara University. In 2013, he had the Professional Certificate of Program in Pharmacovigilance, Catalyst Clinical Services Private Limited, Delhi. 2009, Professional Diploma in Clinical Research from Catalyst Research Clinical Services from Delhi. And 2007, he did his Masters of Pharmacy, Master in Pharmacology from Department of Pharmaceutical Science, Drug Research, Punjab University. In 2004, he did his Bachelors of Pharmacy from Department of Pharmaceutical Science and Drug Research, Punjab University, Patiala, Punjab. So been a member of number of bodies, and especially is member of Secretary of CPCA and CPCA nominated biological scientist of Chandigarh College of Pharmacy. Awards and recognition from Association of Pharmaceutical Teachers of India, Appreciation Award for Promotion of Pharmaceutical Education in Punjab in 2016, Outstanding Research Award for Excellence in Research, Patent and Consultancy Work in Academic Year 2018-19 by Chitkara University, Punjab. He is a registered member of Pharmacists of Punjab Pharmacy Council, registered life member of APTI India, registered life member of Indian Pharmacology Science Society India, registered life member of Society of Pharmaceutical Education Research. He has number of patents to register, almost 12 patents, and he has published number of book chapters and to publish 2 books. Sukhman sir has told me to give you a brief introduction, but you have a vast introduction. So with this, I would like to hand over to you. Welcome once again for this session, and thank you for sparing your time, sir. Over to you, sir. Sir, you are muted. Please unmute yourself.
Hello.
Yes, sir,
Hello.
you're audible now.
Yeah, I'm audible.
Yeah, very much, sir.
Yeah, okay. First of all, I just want to thank Sagar Institute of Research and Technology and Science, along with Ms.Seema, Sharma, the organizing secretary, and Dr. Jitendra, the convener of this particular online workshop on comprehensive workshop on guidelines of CPCSE for animal experimentation. So first of all, already Dr. Shamsher has covered a lot of on CPCSE about its ethics and why we need CPCSE, why we are working on the animals, what are the preclinical studies, what are the in vitro studies. So so that I have cut short my presentation from 100 slides to slides. So starting with the very first one slide. It's audible?
Yes, sir.
Yes, sir. Like, now starting for the very first, what do you mean by pharmacology? Before switching to the pharmacology, it's a basically biomedical research. What do you mean by the biomedical research? In biomedical research, basically we have some pathological conditions. That's why we need animals. That's why there is a role of CPCSE. That's why there is a role of institutional animal ethical committees. Now we have to create the pathological conditions. Then we have to go for the new molecule, whatsoever the molecules are there, new chemical entities are there. Then we check their effects. Like in biological, we divide pharmacology in two sections. One is pharmacokinetic and dynamic. So to create the pathological condition, we need animals. And so for the same, we must better understand the normal physiology and the cause of disease to induce disease in an animal model. Or what's of the drug, what's the new chemical entities there that is developed, like vaccines are there, new drugs are there, like in corona case, there are a number of antiviral drugs are there. To prevent or to treat the disease, one animals. Then we have to test the safety, efficacy, and study of the drugs. So for all these things, we first go for the clinical physiology and clinical biochemistry. Then whatever the response are there, like cellular biology or response of the particular drug. So for the same, there are some novel interventions. Like we have a molecule. That molecule must go through high throughput screening. We have thousands of molecules, but it's quite difficult to perform the preclinical studies on thousands of molecules. So through high throughput screening, we can get a conclusive evidence that these 10 molecules show a very good effect. So to overcome the number of animals that we're using for our study, so one should prefer first go for the in silico study. So in silico study, we go for the ADMT, molecular dynamics are there. So we get the conclusive evidence that this particular molecule, whatsoever the new chemical entities there that have some interaction with the receptors. Once we get the conclusive evidence out of 10 molecules, 2 or 3 molecules have shown the efficacy and affinity with respect to a particular receptor from the in silico docking studies, we should move to the in vitro studies. Like Dr. Shamsher has already covered in very extensively the what type of in vitro studies are there. So I have cut short these in vitro studies. So once from in vitro study, we get the conclusive evidence that one molecule show its efficacy. We get the IC50 value, we get the ED50 values, and it shows potential. Then we switch to the in vivo studies. In vivo study, basically the studies in which we are using the large animal, we are using the small animals. This is one example that I have taken. This is like for a drug that is basically acting on a high fatty, high sucrose diet molecule. Like in this particular, we have the number of molecules are there. These are the new synthetic molecules that are basically have some anti-adaptogenic effects of. They helps to uptake the fatty acids. So first we have the molecule. We go for the high HTS studies. From the HTS studies, we go to the docking studies, and from the docking study we have find some affinity with respect to particular receptors. So the in silico prediction, we get PUR, QR, ARB, studies. Once we get a conclusive evidence that this particular molecule have some affinity for the receptor, then we go for the fibroblast cell. That's an in vitro study, and once we get that particular data that we have used 3-3 L1 cell differential line studies, and we get the conclusive evidence that this particular molecule has some potential, then we switch to the animal studies. Now, why we need animals basically? Because when some molecule, we get some molecule, it's quite difficult. We should directly switch to the human beings. Because we don't have any safety data, we don't have efficacy data, we don't have ADME data of that particular molecule. So we must perform some studies before switching to the human beings. So for the same, we use animals. Because animal shows the clinical simulation with respect to human beings. Although there is a little bit differentiation is there, that's why we are using rats, mice, guinea pig, rabbits, monkey, and dogs are used. Because every particular animal has some simulation with respect to human being. So first, these are the biological similar, then they have some anatomical, physiological, and genetic similarities with the animal, and then they are susceptible to the health problem as the human beings are there. Like if someone is suffering from Alzheimer's disease, he has amyloid beta plaque, tangles are there, and the similar conditions are created in the animals. So we can easily simulate, we can easily get the results, whether parts of the molecule are we using, they are showing, if they are showing some result, if they are basically can overcome this amyloid beta plaque problem in the animals, that they must do same in the human beings also. So second of all, like animal, whatsoever the animal are we using basically in India, basically we're working on mice, rat, guinea pig, or rabbits. These animals have a very short lifespan. So once, like if we want to conduct some chronic studies, like in human beings, we are going for anti-diabetic, antihypertensive drugs for 2 to 3 years. Sometimes it's 5 to 6 years. By what way we get this data? We can use a particular drug for 5 years, for 3 years, for 6 years, basically from the animals. Like in acute and chronic studies, we go for the animals, we give particular drug for 3 months, 6 months, 1 year, 2 years. If there is no toxicity is there, that means we can use this particular drug in human beings for longer period of time, because these drugs don't show as a mutagenic and teratogenic effect in the animals. So by the virtue of these animals, we are able to go for the human beings for the longer duration of time for their respective treatments. And the second, these animals are easily maintained in the conditions, and we go for the controlled environment in animal house as per the CPCSE norms. Now, it's a very, how many animals basically are used in these years? And you will shocked by just by the numbers, like it's approximately 20 to 23 million animals are used throughout the world for the preclinical research. That's why we need CPCSE. That's why we need institutional animal ethical committees, because these animal ethical committees play a very prominent role to cut short that this number of animals, and we should lay down some particular, you can say that guidelines, so that we everyone should remain constrained while working on these animals. Like I've already shared with you, most probably maximum of the works of the researchers are there in biomedical, in pharmacology, in pharmaceutical sciences, in biotechnology, microbiology, virology. They are basically using more than 95% of animal what we're using in research. They are basically rats and mice. Then 4.25 are basically the other animal that are using it. It include rabbit, guinea pig, sheep, fish, frog, insect, and other species, and approximately less than 1%. These are basically large animals. And in CPCSE, we need another facility to work on the large animal like cat, dog, primates are there. So it's a brief, what's the number of animal per day we are using throughout the bird, and you can check it out. And like we are discussed, we are now in this particular while in this webinar, so much animal already used in other part of the bird, like in mice, approximately 8,000 mice per day are used for the biological research and pharmaceutical research. And on the similar pattern, the fish, rat, birds, sheep, rabbit, amphibians are there, like guinea pigs are there. And the number is very much, okay, 1 per 12 seconds. That means a lot. Then a lot of research is going on. So we have to take care about all this aspect while working on these animals. So before proceeding towards the CPCSE, like already Dr. Shamsher has given brief about what are the basic phases are there while we switching from the in vitro data to the preclinical data. Then we move to the, you can say that the clinical data, when we go for the clinical trials, like I have already shared with you. First, we get what are the synthetic compounds are there, and then we test the synthetic compound. Now, in these days, we prefer one should go for the docking studies, molecular dynamic studies. So we get a conclusive evidence from out of 100 molecules, we get 2 or 3 molecules that shows their effect. Then we switch to the preclinical studies, and once we get the efficacy, toxicity data from the particular animal studies, then we switch to the INDA. From INDA, then we switch to the NDA, and then we start the phase 1 trial, phase 2, phase 3, and then that particular drug will be used in the future for the human beings. So as Dr. Shamsher have already discussed this with you, like before filing an investigation or new drug filing, we must, what type of data we need? We need about efficacy data must be there. We need toxicity data. And then we must find out some mechanism, like we have to conduct some microarray studies, proteomic study must be conducted, and like we use RT-PCR, we can go for Western blot, ELISA, and once we get the excipients and we get the active ingredients, once we get the toxicity data, potency data, and we get there are so many bioassays are there, then we file the whatsoever documents we have collected from whatsoever the research we have done, whatsoever we have studied connecting the animal being. Then we go for the INDA filing, then it's reviewed by some evaluators and researchers. Then if they find it out, it's stable to use in the human beings, and then they switch to the animal studies. Now, starting from the CPCSE, first of all, that is a basic topic of the conference, that comprehensive workshop guidelines of the CPCSE on animal experimentation. So like I would love to share, first of all, there is a story. Basically, there is a first time when there is no norms with respect to like CPCSEA through, and there is no norm with respect to the first animal that is used. It's a basically dog, the name Laika, that is basically they have sent into the space to do what type of changes are there once we have sending the human beings into the space. So like after that, once they get it done, then in 1641 they have sent the first time they have sent the cruelty law on the, well, like if someone want to work on animal, they should follow some rules and regulations. That part already covered by Dr. Shamsher in very brief. So that's why I have switched over to the basic things. So now, why we need CPCSE? Basically, we need CPCSE to basically to save the animal rights and for the animal welfare. Basically, it's not about we should basically when we are doing medical research, then it does not mean that these are the animals, they don't feel pain, they don't you can treat by any way. Then there must be some constraint on a researcher, whether it's a teacher, whether it's a research PhD scholar, whether it's a MS students. They should know it's an animal, they have feelings, they have perception, so that we can what sort of the rule and regulations are there, so that we can work in a coherency with that particular IEC and CPCSE norms, so that we can maintain the animals in the animal house. Okay, basically these guidelines are basically provide the human care to the animals used in biomedical research, so that we can we enhance the well-being of the animals, quality and advancement in the biological knowledge that is relevant to the human and animals. So basically, CPCSE basically is a committee for the purpose of control and supervision of experiments on animals. It's a statutory body that is formed in 1960 to prevent the cruelty of the animals. Then it got framed again and again in 1998 when the Maneka Gandhi is there. That will get full-fledged shape, and its headquarters in Chennai. So it has many guidelines and subcommittees are there. So while switching to the CPCSE, there must be some guidelines. That's a good laboratory practices. Good GLPs are basically, you can say, another step towards the CPCSE. Like good laboratory practice was intent to assure quality, maintenance, and safety of animal in the laboratory studies, whatsoever the study we are doing basically. So basic vision that I already cleared by the Dr. Shamsher. What is the vision behind the CPCSE? The vision behind basically the CPCSE is to ensure the animals are not subjected to unnecessary pain or suffering before performing the experiments on them. There should be some norms, whether we go for the euthanasia, whether we go for the analgesics, whether we are performing some techniques. There must be some rules and regulation and guidelines while performing these experiments on the animals, so that we can get the conclusive results, evidence from the animal experimentation. So the basic goal behind CPCSE is to promote the human care of animal used in biomedical research and pharmaceutical research, to provide specifications that will be enhance animal well-being and quality of research. And the objective is basically the basic objective of the CPCSE to control whatsoever the institutional animal ethical committees are there, to regulate their functioning, to provide some, you can say that it's like a body that is coming to your institute that check it out whether you people have a well-maintained animal house facilities there, with all the facilities that are basically as per the rules and regulation of CPCSE. All the member must have knowledge up to that extent of what the members are there. They know about animal welfare, animal well-being. So these are the basic motives of CPCSE. Now, the functioning of CPCSE, as we already know, basically, first we go for the registration of our animal house, like whatsoever the animal house facility, whether it's a centralized animal house facility, whether it's specific to a particular institute. So first of all, we go for a registration to the CPCSE. Then we can, based on the whatsoever the norms are there, like already in the previous presentation, we have, there is 5 to 8 members are there that are actually constituting the institutional animal ethical committee. And then we have to nominate some, and we get the nominee from the CPCSE, we get the link nominee from the CPCSE, and get the scientists from the other institute, from the CPCSE. These are some persons who have knowledge, who have great expertise in to regulate or maintain the IEC of a particular institute. Once we get the approval and we get registered with the CPCSE, then there is inspection. Once the inspection is over, then we get the permission. Actually, there are 3 types of forms are there, like Form B that we will discuss in later on, Form B, Form C, Form D. In Form B, we have to put all the whatsoever the knowledge is there, whatsoever the experiment protocols are there, why we need animals, why we want to conduct these studies, what are the number of animals we need conclusive to get the conclusive results from that. So that's Form B. We have to fill all the information in the Form B. Then in Form B sent to the IEC main nominee, link nominee, what are the members are there, then they go through it, then they file or some, whether there's some by what way they can improve the content of Form B so that they can they can reduce the animal suffering and number of animals that we are using on the basis of scientific rationales. It's not about the human nature that you can do this and that, but basically on the basis of scientific content. Then we get the permission to conduct these experiments in our labs or animal. Then there is some we have some selected vendors are there from where we can purchase the animal. Then if someone is not following these rules and regulation, now in these in today's scenario, the legal actions are quite prominent if someone is not following the CPCSE and IEC norms. So like already in previous time in 2004, there are basically, along with the good laboratory practices are there are 3R principles, but now then they switch to the 3R to the 4R. Then from 4R in these days we are going for the 5R, as but there is a discussion with the, but why we need these 3R principles like reduce, refinement, reduction, replacement? Why we what is the worry behind about why CPCS is basically working on this 5R principle? It's basically it's a moral obligations. It's emotional issues also while we're working on the animals. Now, basically animals are, you can say it's a privilege to work on these animals, but if one say it's my right to work on the animals, it's wrong. So when you have a privilege to do the experiment on the animals, then there must be some issue, like there must be some moral obligation, then there's some government laws are there, then some emotional issues are there for the animal rights and animal well-being. So like already there is a discussion about to reduce, reuse and recycle, then switching to the 4R, replace, reduce, reuse and recycle, what are the animals and are used. Now in 3R, basically with respect to reduction, like already in the, Dr. Shamsher have discussion, being a CPCSE nominee, when we are going to the other institutions for the inspections and we are going through the Form B, there is in many time I have checked personally, I have seen that in the same college, in same department, people are working on a one model, but they need animal. Like if someone is working on anti-diabetic model and they can, there are 6 and 7 persons are also there. They are also working on a same anti-diabetic model on STs that induce diabetes, and they need vehicle, they need disease control, they need, you can say that a positive control, whatsoever the reference drugs are there. Why we need all these 3 animals when the studies are the same? That's the role of CPCSE nominee. That is, here is the role come from the whatsoever the scientists from the other institute. Here is the role of link nominee. They can suggest why not you can share the data for the vehicle control group, for the adaptive, for the disease control group, and that will help in the reduction of the animal. Now, so that we can use properly, like if someone is working on these transgenic animal, these are very expensive animal, and we in normal institute is not able to purchase these animals, and at that time the reduction in animal with proper refinement, proper techniques, and we know what type of you can say parameter we are going to assess. So once reduction, refinement mixed together, there is a reduction of, we easily reduce the number of animals. Then like already there is a discussion about the replacement. Like in replacement, what happened? We can we cannot directly go to the animals. Like I have already discussed, we go for the high throughput screening, we go for the docking studies, we go for the in vitro studies. Once we get that particular molecule have some affinity, once we get the IC50 of a molecule, then we are selecting 10 molecules. But after performing these experiments, we get 1 or 2 molecules that are actually showing their effect up to a great extent. So by this way, we can also reduce the number, and we can also replace the techniques. Like if, like in case of Dr. Shamsher has a very good facility for zebra fish. Zebra fish is quite good if we want to conduct on CNS or whatsoever the neurological disorders are there. And very fortunate, we don't need any approval from the CPCSE or IEC if we want to work on zebra fish. So what's recently replaced his study to the from animals to the zebra fish. Now this one is the 4th one. Basically, in 4th one, we can improve the social life of these experiment animals, basically by professionally and on the basis of our expertise. By what way we can reduce the pain of animals, intellectuals, like in this one. And the 5th one, there is already discussion, the responsibility of researcher to maintain the ethics and integrity while performing this experiment. Like I have already shared with you, while we are working on the animals, it's a privilege, it's not our right. So responsibility as a researcher, as a person belongs to biological sciences and pharmaceutical sciences. So we should work in line with the whatsoever the guidelines are there, good like GLPs is there, CPCSE norms are there. And this is as per already we have discussed CPC, the members that are basically involved in IEC, from the scientific community, regulatory authority, basically it's a main nominee, link nominee, then animal activities are there, they are there, basically it's a, and that particular committee have 3, basically to maintain the quorum, we need 5 members in a particular committee. And CPCSE already divided into two parts. So if someone is, if people are working on animals, then they will go for IEC. If someone is working on viruses, on a recombinant gene technology, then they are going for the institutional safety board. Now, this is something that we have to discuss. Like, while selecting the animal for any experimentation, we must know the life cycle and whatsoever the, you can say that parameters are there. Like we must know about their HB levels. It's a basic, it's in a normal nave animal. So once we know all these parameters, it's quite easy. Like if someone is do the pharmacokinetic study, and this pharmacokinetic study is going for 48 hours, and someone is thinking, I want to work on rat, but it's quite difficult to work on rat. So that we have to switch our study to the rabbits and on the dog, because we know that a lot of samplings are there with respect to blood. So once we have the knowledge of all these parameters, and this will help to switch to the select a particular species. So there are some standard operating procedures while we are performing on the animals, like first of all, by maintaining the SOPs, like you have seen these SOPs in your laboratories also. In animal husbandry, maintenance, breeding, we will discuss in brief also. So that we have already discussed. For small animals, we go for the approval for IEC. For the large animal, there is subcommittee that will basically go for the recommendation SCLEA for the large animals. And IEC composition, previously it's for 3 years. Once we have the approval from CPCSE, now it's for the 5 years. And the main member is the chairman, that is basically head and the director of the institute, and member secretary. One should have the knowledge with respect to animal experimentation. And other member are the main nominee, link nominee, scientists from the outside, socially aware nominee, that are nominated by the CPCSE for 5 years. Now, important activities of this, basically IEC, you can say that, or CPCSE. First of all is the registration for experimentation. It's basically a facility created by a particular institute. Then once a particular facility is approved, then we get the permission to conduct the experiments on the animal. And the third part is we should also register for the breeding or the trading of the animals if we have a sufficient space for the same and sufficient expertise to go for the breeding and trading. So we need veterinary care for the animals. Like why we are going for a quarantine? like there are 3 stages with respect to one is quarantine stage is there, then stabilization stage is there, then separation is stage there. Like once we received the new animals from a vendor or CPC registered vendor to our own institute, then there are some psychological, physiological stresses are there. To overcome these psychological, physiological stresses and to stabilize the animal before we go for our studies, and that's the most important part. One should give some time, like you can range from 1 week to 2 weeks, sometimes it's from 3 weeks also, once the animal get normalized, and so it depends upon that transportation also, okay, by what way we are transporting the animal to our own, you can say that in our own facility. So once the quarantine phase is over, then we basically stabilize the animal, then we go for the separation process. It is basically why we go for the separation process physically, just to eliminate the if there is some to eliminate to halt the transmission or to eliminate the anxiety, psychological behavior changes are the interspecies conflict. Like you can't put a rat and mice together in one cage, so physical separations of the animal are very important. And basically whenever we are going for the physical separations, like we must do it in a particular, like we have whatever the animal facilities are there, we have a dedicated facility for mice, rat, guinea pig and rabbits. We can't intermingle each one into single room, like we're putting the rat in a single rat, mice in a single room. We must have dedicated facilities for the same. And the experimental area, what's the experimental area, should be clean first of all. If we are going for some surgical procedures, then it should be, you can say it's basically aseptic area, and surveillance must be there. While in these days, it's a mandatory requirement, while you must put some cameras in experimental area and in animal house, so that we can have surveillance whether what type of experiments are we doing, whether we're doing these experiments in the line with the rules and regulations set by the CPCSE. And personal hygiene plays a very prominent role while performing these experiments, and treatment of animal, like once we like if someone is doing some studies and some selected animal on stroke model, then we go for the occlusion of carotid artery, and once we occlude the carotid artery, then we have to sew the animals. So post-operative and intensive care plays a very important role for the rehabilitation of the animals. So one must have expertise while performing these experiments, and it's duties of I can practically say it's a duty of member secretary and animal house in charge to just check it out whether everyone is performing as per the norms laid down by the CPCSE. Now the physical facilities are like it must be have some concrete building is there that is actually we can maintain the temperature, we can maintain the light, we can maintain the moisture. So we must have all these facilities, automated facilities that can actually you can say we can daily note down the relative humidity, we can daily note down the temperature, we can daily note the, and these days it's quite it's a you can say that it's a mandatory to have automatic switching in the animal house, that after 12 hours there is switching of light, then we don't need someone physically present there in the animal house to switch off the light and on the light. And and the corridor, there are like dirty corridor is there and there is clean corridor is there. And I have seen in many institutions, they are using same corridor for the movements are there with respect to instrumentation animals, and that is not as per the norms and regulations of CPCSE. So we must have a dirty corridor, one should come from one side and go from the other side. So we must maintain the dirty and clean corridor. And the animal room should have, like in these days, it's approximately 90 lakhs light must be there, so that can get exposure just like a daylight. And the floor should be smooth and moisture proofs are there, and we should maintain the aseptic conditions in the particular parts of the rooms are there, proper drainage is there, and we must have a dedicated facilities for the you can say for the animal bedding, for the feed, for the cage, for the washing, whatsoever the things we are using in the it should not be placed in a single room. No, environment, that is one of the most important point when we are going for the storage of animal. Like we should, like it's basically as per independent, one should maintain the temperature in the range of 18 to 26. But as per CPCSE, it's 18 to 29 degrees Celsius. That's quite sufficient. That's why we need ACs and we need centralized cooling system in the animal house facilities. And then relative humidity, one should maintain the relative humidity that from 30 to 70%. And that's why in these days, whatsoever the nominees are there, they need automated temperature and humidity controller so that there is no physical variations are there. And ventilation, as we need 12 to 15 air cycles per hour. And very frank to you, it's quite difficult to maintain all these things in the normal conditions. But as per the norms, now it's mandatory for whatsoever the institutions, whatsoever the facilities are there. And like I've already shared with you, there must be 24-hour power supplies is there, then lighting system must be there. We must have emergency backups like generators are there, and noise control. This is one of the most important point while we're working on the animals. Because if there is a noise in the animal house, that will actually create anxiety and that will actually register a release of catecholamines. So we have to maintain an animal house facility. There should be minimum noise or totally noise-free environment around the animals. Now, animal husbandry, like there must be set norms. We can use this number of animals in a particular cage, like as per my expertise, I am always preferring we should go for to 6 to 8 mice in a one cage and 3 to 4 rat in one cage, so that we can maintain a you can say that we can maintain an environment for the animals, so that they can easily access to the foods and water, and they have sufficient space for their, you can say that they have freedom of movements, they have space, and there is no psychological and you can say physiological barriers in particular when we're, and the bedding is play a very important role. In previous times, the people are using husk as a bedding material, but in these days there is specific bedding that one can use for the you can say that for the animal house. And then food and water. Food deprivation is not there, but most important, whatsoever the food material like pellets are we are using, we are not going to use the normal pellets. They must be procured from the whatsoever CPCSE has given a list of some you can say some vendors from where we can get the food pellets for the animals, and that particular have a, you can say that a particular ratio of amino acids, proteins, and we must get a, you can say that certificate of analysis whatsoever the you can say pellets and food material we are procuring for the animals. And like in these days, when you are going for the first inspection, like if somebody is going for the animal house inspection, like they want certificate of analysis whatsoever the feed we are procuring for the animals, so that they have whatsoever the like content in form of protein, amino acid, they must be having a particular ratio that can actually maintain minerals, carbohydrates, and like feed does not contain basically insecticides and hormones, any type of antibiotic, any potential environmental toxins, heavy metals. So that's why we need a certificate of analysis for the whatsoever the material we're using. Like I have already discussed previously in these days also, there are many institutes that are using the bedding, but recently CPCSE have given that use pellet bedding that can easily absorb that urine material, so that if we are using husk in the cage for the animals, that once after they urinate or what's the fecal matter there, they get sticky and they have a offensive smell, and it get you can say totally provide a wet condition that actually affects the psychology and behavior of animals. So so bedding should be as must have some absorbent nature, and it's free of you can say that. That's why if someone is using husk, then he must autoclave the husk first, then he or she can use in the animal house facilities. So and one should remove husk if someone is using husk, like one should remove husk periodically after 1 or 2 days and adds fresh material, and if we are using husk, it's quite difficult. Okay, we can use husk twice a day, but if we are using absorbent bedding, that's it quite easily. You can maintain the animals for a week, and you can change the bedding 2 times in a week. But if you're using husk, then I should prefer one should go for 3 to 4 time change of bedding in a week, because it's quite difficult to maintain the hygienic conditions with the husk. And then sanitation and cleanness, that is very important point while we are taking care of the animal house facilities. We have already, there must be some sufficient space for the corridors, whether it's a dirty corridor, whether it's a clean corridor, and we should provide aseptic conditions in the animals. We need to go for fumigation also. We use the disinfectants, detergent to to clean the floor. Then waste should be removed regularly and frequently, and cages should be sanitized before using ones. Like you can use any type of sanitizer. What are the facilities we have? We can use ethanol, isopropyl alcohol to sanitize the alcohol. Then the most important point. This is the this point is already discussed by the, you can say that Dr. Shamsher also. What type of you can say that anesthetics are used, or what type of euthanasia procedures are there we can use during the experimentation. Like CPCSEA has provided a list of you can say that list of anesthetics that are used in laboratory animals, like ketamine hydrochloride is there, and there is a specific dose for ketamine hydrochloride for mice or rat. One should go for in the range of 20 to 24 mg per kg dose. Then we have phenobarbital, thiopental, sodium urethane. These are the commonly used anesthetics in the you can say in the animal house facilities. Now, euthanasia basically it's a merciless killing. After we're doing the experiment, we have to sacrifice the experimental animal, but the procedure must be painless killing. So there are 3 to 4 methods are there. We can use we can go for the overdose of anesthetics, like barbiturate overdose is there, chloral hydrate overdose is there, ketamine hydrodose is there. We can sodium pentothal high dose is there. As per my expertise are there. And like for the physical method, after this painless killing, like I have already discussed with you, we should provide, whenever we want to transfer the animals from another facility to our animal house facility, we should maintain the environmental conditions accurate as per the use. Like we should go by the AC, what sort of the AC cars or AC, like you can, what sort of any facility that have maintained the temperature. And then number of animal used per kg, it's one of the most prominent, and space per animal in the animal house. That is actually helps to calculate the animal house facility. Most probably for one mice we must have a 20 to 25 centimeter square space, and for rat and mice we need 100 centimeter square space. And along with as the animal grow, we like for a rabbit we need 1000 centimeter square space. So once we have once we maintain all these facilities in our maintain all the facilities in our animal house, then we can think we can do the experiment in a better way, very frankly. So like preclinical trials are there. So what is a preclinical? These are the preclinical study. Once we get a compound that is showing its efficacy in the in vitro or docking studies, then we switch to the preclinical trials, or you can say that non-clinical trials. These are the basically new drugs substances that are we injecting or into the animals, and then we're checking out whether it will show its effect or not. And along with this, once we get the efficacy, then we have to check it out. There's a safety profile also, whether this particular molecule is safe, and by performing the acute toxicity, subacute and chronic toxicity studies for a longer duration, whether if once we're switching from preclinical to clinical studies, whether this particular compound we are using for a longer duration that is showing some toxicity or not. And products may also include, we can use new medical devices like some insulin devices. We are actually working on some insulin device, and we're using animals where by how much insulin is delivered in the animals, like gene therapy. For this type of you can say experiments, we can also use the you can say we can also use the animals. So like in daily routine life, we are basically using the rodents like rat, mouse, guinea pig, hamster, and once we actually successfully industries, basically once they get the conclusive evidence from the small animals, they switch to this result to get the conclusive evidence to the large animals like cat, dog, monkeys are there. And if we get the, you can say that as per OECD guidelines, these are the guidelines, these are the universal guidelines. While we are performing some toxicity data, we must have data from the 2 different species in case of rodents. And once we get the conclusive or safety data from the 2 different rodents in the, you can say that from small or large animal also, then we can go for the clinical setups and clinical studies. So like that we have already discussed, once we have identified a drug target, then we develop a bioassays, then we screen the animals, and once we screening is over, then we go for the find out the efficacy and potency, then we filing the investigation new drug application so that we can switch to the clinical studies. And these are the you can say that whatsoever these are the parameters screening test, like CPCSEA always prefer whatsoever the toxicity study we are performing, one should perform as per the OECD guidelines, whether we are going for the oral toxicity study, parental toxicity study, when we are going for the short-term treatment, long-term treatment, one should follow all these things. We should need a conclusive evidence for all this, from all these whatsoever the experimentation procedures are there. Like we have to do for the in vitro studies, we go for the bacterial culture, like then we have to work on the different animal models that are clinically similar to the human being. Then we go for the, we just try to find out some system biology, like system pharmacology in these days quite prominent area with respect to researcher. Like if we are working on a drug that we're basically watching it act on a central nervous system, but once we are injecting a drug that can also affect other part, whether it affect on GIT. And once we get the conclusive evidence, then we switch to the, if we get the, and now in these days what happened, we get conclusive evidence from the preclinical data, but these drugs get failed when they come into the human beings. Why it's happened? It happened due to system biology. Because basically, previously the scientists are not giving so much preference to the system biology, but due to major failure in clinical trials, like in phase 2, phase 3, even phase 4 too also, it's only because we are not checking out the system biology properly. So in these days, scientists are working exclusively on system biology once we basically finding the safety and efficacy data. So like once that we have already discussed, once we get the data from the preclinical, then we switch to the IND and go to the, like, this is the most important slide in these days. While like in India, people are working on so many compounds, like even some herbal extracts are there. And as per OECD, whether like OECD, as per in Indian Ayurvedic system says all the plant extract what we are using, they are safe. But OECD says whatsoever the molecule we are using first time for any condition, we must follow some you can a stepwise procedure. If there is no data available for a particular molecule, then you should directly go for the 300 mg per kg dose to the animal. Just find it out whether there is some mortality is there, whether they have some behavioral changes are there. If there is no behavior changes, there is no mortality, then we can say it's a safe. But and as far as if we are using plant material, we can go up to 5,000 mg per kg. And if we are using some organic material or synthetic material, as per OECD, we can inject only 2,000 mg per kg in the animals. We are not going behind 2,000 mg. It's as per the OECD norms. And to perform the acute and chronic toxicity study, we are going for the 3 to 4 animal, and that's quite sufficient for the statistical significance. So like this we have already discussed in previous slide. In preclinical studies, we go for the isolated in vitro studies, we go for the synthetic and non-synthetic reaction study. When we are going for the metabolic studies, then we go kinetic and bioavailability studies, and that's why we are using some softwares and to find out the pharmacokinetic studies. Now, what are the limitations of the preclinical studies? And that is the major reason behind when we're performing these preclinical studies. And there is you can say that there is a failure of drugs. There is a failure of drugs basically when we're using the same drugs in the human beings in different phases, like basically suitable pharmacological model. That is one of the most prominent model. Like I want to share some examples with you. Like if someone want to work on a conclusively on, you can say that Alzheimer's disease, there are different models are used, like we can use by STZ model. There are some drugs, calcium channel blockers are there, because scopolamine is there. But on international platform, people should prefer, we should go for the amyloid beta 140 and 42 plaques. We directly inject through ICV or stereotactic apparatus into the brain, to the mice or rat brain, so that we can get the very simulated conditions in the animals. But when we are going for some other, these molecules also generate amyloid plaques. Does not mean that if we are going for STZ model, it does not form plaques in the, but we must stick to the particular rationale. We need, we are working on Alzheimer's only. On the similar conditions, like Parkinson, there are a number of Parkinson's models are there. Like Nature Science have given a very descriptive descriptive paper on why one should go for MPTP model as compared to rotanin model, or other environmental toxins are there, and what type of doses we can use. Actually, you can find a number of, you can say that number of animal models, and number of doses are there of particular compound with respect to chronic and acute levels. But when this, then there is a problem with the reproducibility of data. Like I'm sorry to say, the 95% of Indian researchers paper don't have any reproducibility, and the basic reason behind there is no reproducibility, and it's a conclusive evidence given by the Nature Science. The 95% of Indian paper don't produce reproducible data, and the reason behind it, there is, you can say there are a lot of ununiformity in the animal model. We are selecting the animals, we are selecting the dose. So we should follow some international norms while performing these experiments. And that's the only way we can get the conclusive evidence, and someone get the, you can say, reproducibility of your data. And then toxicity testing, basically we are not performing toxicity testing, like we are going for LD50 if you are finding some papers, but nobody's bothering to perform the studies for 6 months and going for the chronic phase to 2 years. It's a time consuming process. If we are not able to perform these studies up to, that's the only reason we are going for LD50, and when these particular compounds come into use, they don't show their type of result, because we don't done the toxicity studies up to 6 years, and you can say 1 year, 2 years. And the main constraint again comes a large number of animals we must need, but as per the norms, like we should be strict on the numbers. As a CPCSE member, it's our first duty to protect the animals, so that we can save the rights and of the animals. So then extrapolation of the toxicity data, that is a little bit, you can say we can discuss whole day on the extrapolation of toxicity data from the animals to the human, and you can find there are a lot of glitches out there while we're performing all these things. And like ADRs or AE, this is one of the prominent reason we are not able to find it out in the animals. Because when if we're using some compound, then we have to do a conclusive. We go for the whole biochemistry, whole histology of every organ, biochemistry for the whole organs. Then we have to check it out, all the behavior parameters, so that each and every aspect we should cover when we are performing the preclinical study. And it's quite difficult to perform all the studies in a single, you can say that in a single shot while performing the studies in animals. Now, this is on my last slide. You should take care about the lab animals. After all, you will owe them. This one is for the just to in the last while there are MS, MPharm students are there, PhD students are there. What are the important point while we are selecting animal model that we are using in our thesis work or doctorate work? And that actually plays a very prominent role. First of all, number of animal. It plays a very prominent role. Like if someone is working on stroke model, someone is working on diabetic model, and you should be strict on whether how many animal are you required. And basically, when we are working on a particular model, then we must know like if someone is working on stroke model, there is a mortality rate of 30 to 40%. If someone is working on streptozotocin induced diabetic model, there is a mortality of 30 to 40% as per the literature. But as per there is some new guidelines, are stroke guidelines are there, diabetic guidelines are there, American Association of Diabetes. Now, if we are follow these guidelines, then we can decrease the number easily. So selection of animals plays a very prominent role. Like there are some studies we can't use female animals than because the sex hormones plays a very prominent role with respect to whatsoever the animal models are there. So we have to find it out every glitch in the study key, how many animal we are required while designing any type of protocol. And weight ranges also plays a very prominent role when we are performing some AD studies are there. If you can say we can use a younger animals, we are not I have seen personally in many studies people are not checking out the weight and age while selecting the animal. We don't thinking like we just need animals, we have compound, we want to do the study only. But age and weight, they play very prominent role when we are getting some results. So one should stick on these things very carefully. And like I have seen many papers where the like we have used a vehicle control group, that is a neighbor group you can say. Then we have used a disease control group, and there are a lot of studies that I have gone through. There, people don't use the standard and reference drugs. One should always use a standard and reference drug so that we can compare our result with whatsoever the reference drugs are there, standard drugs are there. And then we have to prepare a treatment schedule. Like if we are working at that particular day, we are going to sacrifice the animal. That particular day, we are going for the biochemistry and histopathology. And on that particular day, we're going to assess all the parameters. And with this, thank you. I'm totally here to solve your queries. Thank you.
Okay, so thank you very much, sir, for your nice presentation. Gurjeet, sir, kindly you stop your sharing so that we may start sharing now. And now I open the house. Yes, sir. Thank you.
Well, I'm just stopping the video. Yes.
Yes, sir. Thank you so much, sir.
Okay, dear.
Sir, don't stop your video. You can switch on your video. Stop the sharing that you are doing right now. Right, sir? Yeah. So now I open the house to all the participants. If they have any queries, they can raise their hands so that I'll mute them one by one, and we can continue the session further. Any queries, please? Yeah. I have a query.
Yeah.
I'm unmuting right now. Please name yourself and ask your question now. The username shows admin.
Hello, is it audible?
Yes, ma'am. Please introduce yourself and ask the question.
Sir, myself, G. Sumitra, calling from Tamil Nadu. I'm an associate professor in the Department of Pharmacology.
Yes, ma'am.
Sir, kindly tell me about the rehabilitation technique which you are following in your laboratories, sir, for animal usage.
Rehabilitation basically like it depends upon what type of study we are performing. And like if we are doing some behavior studies are there, and you must before going for the rehabilitation studies, you must know the age of a particular drug, how much wash period is there. Like we are doing the behavior study and we're not going to sacrifice any animals, so we know that this particular drug will take this much washing period from the body. Then you should put this animal into the rehabilitate phase for 21 days. Then you can use it. Like if we are using some surgical techniques are there. But if we know after doing these surgical techniques, we no need to for sacrificing the animals, and so that we get rehabilitate. But we have to maintain all the aseptic things. We go. We have to given the whatsoever you can say antibiotics are there and analgesics are there. You should when we going through the value of 300 pages of CPCSEA. For the rehabilitation, they have given the selected analgesics are there, and you should follow the aseptic conditions. Once the animal you washing it produce hello.
Yes, sir.
When the washing period is over, and once the animal like if we are doing some surgical techniques are there, and but we are not going to
pardon me, sir, is it surgical technique is available to apply for the rehabilitation, sir, like laser technique?
I will share with you example. Like if we are doing some hypertensive studies and we have just want to check it out, there are two type method. One is invasive and non-invasive method. And non-invasive method quite difficult. We use tail and cuff apparatus, and tail and cuff apparatus will cost about 8 to 10 lakhs. But if we go for the normal you can say that biopack instrument, it's cost about 1.5 to 2 lakhs. And then we have to put a probe into the carotid artery. to get the systemic and blood systemic and diastolic blood pressures, if we're not going to for the biochemistry or any other studies. So by doing by this way, once we have to put off the carotid, then we have to stitch again, then we have to wait for the animal that they get healed, and then we can rehabilitate again, then we can use it again. But in behavior study, we have to wait for how much time it will take for the washout period of a drug. And once the washout period is over, and then we can use after 21 to 28 days this animal for the other studies.
Okay, sir. Thank you, sir.
Thank you, sir. Now I open the mic for Mr. Devendra Shirode. You can now ask your question.
Yeah. Good afternoon, sir. Thanks for nice presentation. Here I want to ask two things. First, sir, now PCI restrict use of animals in the B.Pharm college. So how can
Yes,
we
sir.
use experiment? And second question, sir, for cytokines activities,
Yes, sir.
ELISA or PCR, which is better?
Basically, I come to your first question. And the first question is with respect to uses of animal. Like this particular condition is, you can say, universal for all the institute. But if you have PG program, like we are using in the same way, whatsoever the experiments, like whatsoever the thesis part is going for the PG students, we should enroll our students, bachelor student with the PG student should go to the PG lab. Whatsoever now it's depend upon the whatsoever the teacher are there, by what way we can manage. And in our institute, we can send us to PG UG students, B.Pharm students to the PG class. They are performing this experiment so that they can get familiar with. In these days, what happened basically, like it's in the consensus with your question. What are the students are coming after B.Pharm from pharmacology? They don't know about anything about animals. And they have faced a lot of problem while handling with the animals. Basically, the institutes are strict on the, you can say that rule and regulations set down by the PCI that one can't use these animals in a UG program. But PCI don't say you can't use. Your PG program are using the animal. You can send your student to the PG classes. They can check it out by what way the PG students are doing their experiments. And they can also learn to learn the techniques and by what way they can handle the animals. So it's the only way. Recently, we have put some requests to the PCI. We need. We just want there must be some relaxation for the experimentation on animals in case of B.Pharm students. Otherwise, it's quite difficult for a particular branch like M.Pharm pharmacology. The students always have a restriction in their mind. I'm not able to use the animals. I'm not feeling comfortable in the animal, but I love pharmacology. So this type of queries and questions are there. It's only when we are not giving exposure to the animals. So as per my expertise, as per my suggestions, you can send your student to the PG class, so that they can watch it, they can know by what way someone is doing experiment on elements. In PG, we do the thesis in a very conclusive way, like if we are doing some studies, and these studies are basically 1 to 2 months long. So it's the only way. And in second case, if you like in these days, in case of corona, like we are using cytokine storm is one of the major parameter. And on my perception, you can go for RT-PCR. That will provide you a conclusive evidence about the ELISA provides you can say that it's basically you can provide a quantitative data that this much nanogram per mg, you can get the data. But as per my expertise, one should prefer go for the Western blot, and not on the Western blot, you can go for the RT-PCR. You will get a conclusive evidence and no one can challenge your results. In these days, if you are going for the Western blot, now the whatsoever the journals are there, they have changed the norms. Basically, previously the people manipulate the Western blot data. Now, journal need 3 picture of a one time 3 snapshot at once at different time interval with the tagging when you are going for a publisher paper, so that manipulation, one can reduce the manipulation. So I should prefer you should go for RT-PCR if you want to check it out the cytokine storm.
Thank you, sir. Thank you.
Any other queries? Hello?
A participant? Have you any question? Please raise your hand. उनको बुलाना घूमना. Hello.
Yes, Dr. Nilesh.
Yes, sir. There is some network issue, sir.
Don't worry. Hello, Dr. Balraj. बस I'm on the way, but this one. Bas, don't worry. Give me just 5 minutes more. Okay. जी रवि सर बस फाइन और मैं एक्चुअली थोड़ा मीटिंग सीगा दसो प्रॉब्लम इज देयर क्योंकि ना इन जो अपना नेट वाला फाइन है उसमें ना जो
Thank you, Gurjeet, sir. Sorry for the internet. Sorry for the interruption. Actually, net connection is very poor in our area, so no problem. There is a nice presentation, and I would like to thank Gurjeet Singh sir for their valuable time and presentation. I hereby share a token of appreciation on behalf of SIRTS-Pharmacy, just for you, sir. Just share in the screen. So, well, dear friends, that event like cannot be happen because of this renowned speaker, and we are continue our the session on 1:45 after the lunch break. So please join us with using a same link. Thank you for all, and with the permission of all dignitaries, I am going to end this session. Thank you, Gurjeet sir, and thank you, Shamsher sir.